Hello, I'm Rich Whitworth, Content Director of The Medicine Maker. I'm here in Cytiva's Uppsala facility in Sweden for a special discussion, process intensification, not complication. I'm even more delighted to be joined by three experts who are, it's fair to say, far more qualified than me to discuss this topic, so thank you for joining me. Before we get going, I would like to introduce these three experts. So first of all, we say hello to Ruth de la Fuente, Global Products Manager at Cytiva. Ruth is a well-rounded professional with over 15 years of experience in the bioprocess industry. Her background covers process development and equipment design for batch and continuous processing, as well as managing technical transfers and facility designs for commercial operations. Is that correct? You're not on the stand, but we're best just check. Ruth's approach to designing unit operations is characterised by a holistic perspective, considering platform technology, process conditions, facility integration, and user experience. With a focus on customer needs, she strives to provide flexible and cost-effective solutions. Finally, I can say that Ruth holds a master's degree in chemical engineering, as well as an executive MBA. Ruth spends a good deal of her time supporting customers in process intensification strategies and the adoption of new technologies, so ideally placed for this discussion. Next up, Andreas Castan. As Strategic Technologies Leader at Cytiva, Andreas provides leadership and support to Cytiva's bioprocess business. His main area of expertise is in upstream processing, and we'll be talking quite a lot about upstream processing. So again, handy to have you on board, Andreas. Over the last 20 years, Andreas has held several positions within biopharmaceutical development, including project and line management, as well as manufacturing. Before joining Cytiva, Andreas was Director, Upstream Development at Swedish Orphan Biovitrum. Welcome, Andreas.
Thank you very much.
And last but by no means least, we have Nicholas Youngnelius. As bioprocess modelling leader at Cytiva, Nicholas works with the customers, industry associations and internal stakeholders to assess impacts of various process technology options and provide guidance in wisely choosing between these options to meet manufacturing objectives in terms of process economy, facility fit and environmental sustainability. And we'll also be touching on sustainability a little later.
Yeah.
Nicholas also holds a master's degree in chemical engineering and brings 25 years of experience from various companies and positions in the life science industry, half of which was served in different strategic positions at Cytiva, or as it was formerly known, GE Healthcare. So, let us begin our discussion. Before we get into the meat of the discussion, I thought it'd be, I use the word fun, maybe that's too strong. I thought it'd be interesting for you to describe to me what process intensification means to you in your own words. Andreas, can you kick us off? What does process intensification mean?
For me, process intensification is about doing more with less, producing more product, producing cheaper product with less kind of raw materials, facility footprint, whatever it is.
Thank you. Ruth?
Yeah, my definition is not so far from Andreas. I add a little bit that we work differently. So there's a different kind of approaching the same problems and the same challenges, doing more with less, more effectively, but slightly different, and that's where the challenge lies.
Thank you, Ruth. And Nicholas?
Yeah, I think we're aligned so far, because doing more with less would be the first thing I come to think of as well. And that can help you reduce the footprint of the facility or more efficiently utilize your raw materials, all to help you drive down facility footprint and cost of manufacturing.
Fantastic. So we've sort of given ourselves an outline there. Now, to frame the topic of discussion a little bit more, I'm going to turn to you, Nicholas, so that we can frame the topic a little bit in more detail. Andreas and Ruth, feel free to jump in at any time. This is a discussion, so if you agree or disagree with Nicholas, feel free to step in. But first of all, maybe you can talk to us a little bit about the drivers for process intensification. We've already talked a little bit about cost, doing things more efficiently, but can you walk us through that in a little bit more detail, please?
Yeah, I think when we ask our customers, what are their main strategic initiatives and activities that they are working on, what comes out in top almost every time is around improving product quality, improving speed to market, but then also several points around how to manufacture things more efficiently, increase throughput of your facility, and also reduce cost of goods. And I think those are areas where process intensification fits perfectly. And perhaps also an upcoming driver that we see a lot more and more that was not very present only a few years ago is around environmental sustainability, as we alluded to earlier here. And I think that is also an area where process intensification can do a lot of good.
Thank you, Nicholas. So now, delving into that in a bit more detail, I thought it'd be good to go through the sort of the what, when, and why of process intensification on a high level.
Sure.
Can you walk us through those key questions? You know, when do we do it? What do we do? Right, there are many options available, and we'll delve into some of those options in a little bit with Andreas and Ruth. But can you walk us through some sort of high level examples?
Yeah, I prefer to start with why.
Okay.
You need to be clear on what are your objectives, what are your needs, and what are you trying to achieve, and what are your goals with the intensification of your process. And once you have set that, you can start thinking about what. There are different technologies that are appropriate for to address different needs, and I would say no customer is the same. They are all individual. So I would say all our end users have individual needs, and what technology may suit one better may not be the best option to another. So a lot of my time I spend with our end users to discuss and help them identify what are the implications of using different intensification options. So what you should consider in this is that some technologies may require you to use perhaps a new technology that can introduce risk to your process. It can introduce complexity that you're not used to. And these implications may cut across large parts of your organizations. You need to align your stakeholders to be aware of those implications and have mitigation actions for that. But once you have that and are prepared to address those challenges that may come up, you can have a lot of benefits from it. And then when is also a good question, because if you have a technology option that is perhaps more complex in implementation, requires more process development activities around it, or introduce risk to your process as you do things perhaps differently than you have in the past, you may consider when to implement this in your process. Should you do it already from early preclinical work or during clinical development, or even should you wait until you have your product launched and introduce a second generation process? I think that's a trade-off that much depends on your manufacturing volumes and your drive to come quickly to market. It may be a good option to start with a more robust and simplistic process that doesn't require too much activities and that you know works well to get through the early phases to ensure that your molecule is not failing during clinical development. And also you may even want to wait until you're on the market to see that there is actually the demand you're planning for this, and then launch a more efficient second-gen process.
Anything to add there, Andreas or Ruth? He's given us a very good overarching view.
Yeah, I mean, I do agree. Sometimes you can jump directly into an intensify process at commercial scale. It's more challenging, in my opinion, because there's more at stake when you are operating in GMP. I think it's also good to consider it for clinical, because the benefits of an intensify process is that with a small footprint, you can manufacture a lot of product that otherwise will take you a bit longer to make. And it's also good, I guess, to work on our development scales on the platforms that you're going to then later on implement in commercial, because then you get acquaintance with those kind of strategies and you start thinking of those strategies in different ways, because implementing a batch process, it's a different ballgame than implementing a continuous process, and you need to understand where to focus on each one of them.
Absolutely.
What I could add, and what I see when I meet customers is the companies that are really spearheading the implementation of process intensification are the large biopharmas, like you said. When designing a new molecule or a new process, they think process intensification from the start. It's the CMOs which want to match their customers' needs, and it's very much the biosimilar companies which are really chasing every dollar that they can to.
You do see, and we've seen customers that say, we do have our platform, and we may start with a simpler platform, like a batch platform, and then decide if we see, as Niklas said, that the demand is going to increase, we'll move to this other platform, which is more cost effective or has higher productivity.
Great, thank you. So with that in mind, we also said we would discuss a little bit about manufacturing strategies. What are the considerations for manufacturing strategies when we're thinking in terms of process intensification? I guess that's kind of the where, I suppose, in a way.
Yeah. I mean, you need to plan with the end in mind. So what are your capacity plans for your product, and how are you going to scale this product up to the final production scale? Do you need a distributed network? If so, you need to consider how are you going to manufacture this? Are you doing it internally on your own facilities? Do you have a network for that? Or are you going to transfer this to a CDMO? And with a more complex manufacturing process, I mean, we see with intensification, we get typically more automation. We have a lot of more data coming both in and out of the process in order to help optimize the process. You need to have a plan for how you're going to transfer that information to a CDMO in the first place, if you're using that, but also how can you leverage the data that is generated in the process during the manufacturing at the CDMO internally if you want to bring it back in at some point. Can you get the data to benefit for the process?
Great. Any comments on manufacturing strategies before we dive into?
Yeah, I think the point you made on portability is very important when you consider going to CMOs or having a large manufacturing network. You need to design a process which can be transferred in this network, and I think that will kind of dictate or limit your possibilities of the kind of solutions we will discuss later on.
Great, thanks. So we've talked about the why, the when, the what, and the where. I think now's probably a good time to delve into the weeds a little bit with the how. So Andreas, you're going to sort of walk us through, I guess, 4 examples. There's a lot that we could cover. If we had a 2-day workshop, I'm sure we could fill that time as well. But we have a limited time, so we're going to walk through 4 examples, and we're going to do them in sets of 2, I suppose. So perhaps you can kick us off with the first 2. That's N1 perfusion and concentrated fed batch perfusion.
Let me actually start with a kind of high-level perspective. Sure. I think before going into the details and looking into the unit operations, I think it's important to ask yourself, for example, is my cell line good enough? Is my media platform performing well enough to achieve my needs? It's a huge difference if you have a process that is giving 3 grams per liter or 6 grams per liter out from your normal fed batch bioreactor. Secondly, I think it's important to think about what is actually the bottleneck in my process. For example, if my downstream train is idle 50% of the time, the simplest way of intensifying and increasing the throughput is just buying more bioreactors and increase your capacity there. So just having that big picture in mind and thinking about that before going into the detailed solutions for upstream, I think is important.
Do you have some sort of high-level view you'd want to share before we delve into the details?
Sure. I mean, high level for downstream is we are a little bit slave of the upstream. And I would say, I mean, I do agree with you need to maximize your capacity utilization. right? And it needs to make sense for whatever is coming to you. So there's no one, it's like, there's no like, oh, we use continuous always for this, we use batch always for this. There are guidelines. So high level is you can focus on being cost effective, you can focus on maximizing capacity, which is usually what is done in commercial. You can focus on maximizing productivity. You need to have clear what are the limitations of your facility if you're retrofitting from a traditional batch to a continuous, or what are your drivers, what is important for you in that case. And then you move around the options that you have to select the best that you can do.
Nicholas, do you have anything to add to that sort of high-level overview?
Yeah, I think I can underscore perhaps Andreas's comment around the titer. I mean, there are a few levers that are extremely powerful to improve the productivity of your process, and titer is definitely one of those. So if you can spend some time optimizing the productivity of your cell line, that is typically money well spent. Also, production scale, very important. If you can manufacture in larger scale, that improves the efficiency in your process.
Thanks, Nicholas. Now we can
Now
we can dig into the details.
Now we can deep dive in.
Thank you for keeping me on the straight and narrow, Andreas. So kick us off then with the first two examples, and then we'll ask Ruth and then Nicholas to hop in.
Exactly. So you can do things. Very brief comment on the seed train. You can bank more cells. You can bank them in vials or in bags, and basically omit stages in the seed train. You can run perfusion, N-1 perfusion in the seed train. But looking at the production bioreactor, when you start with a traditional fed batch bioreactor, you can basically move part of the proliferation phase out from the production bioreactor into your N-1 bioreactor that you run in perfusion, which means that you cut out 3, 4, 5 days from your fed batch process. We call that a high seed fed batch process because it's inoculated with an N-1 inoculum at a higher seed density. Decreases the time, as I said, but very often we see also that you improve the productivity of that process. Very simple solution to implement as all the technology for N-1 perfusion is available, and you basically stick to your normal fed batch process. The next level of intensification, considering a batch mode, would be a concentrated fed batch, which basically is that you operate a perfusion with an ultrafilter in your bioreactor. You bump up the cell densities, increase the productivity, typically increase the titer 3-5-fold compared to the fed batch process. At a kind of acceptable level of media consumption, and the advantage that you still have a batch process in the capture and downstream. However, higher load of cells, higher load of host cell proteins.
Will I get more cell debris?
Yes.
So that's a good point to bring you in, Ruth. Maybe we can talk a little bit. These are sort of, I think we're ordering these, aren't we, in sort of ease of implementation in a certain way, or barriers to implementation. But now's a good time to bring you in for the impact. You already said that downstream is somewhat a slave to upstream. So what does this mean? What does this mean for you, and what do we do about it?
What it means is we need to catch up whatever is coming from harvest, right? What I ask about the cell debris, because cell debris is something we do not really like at clarification. It makes clarification more challenging, or requires larger areas or different type of filters and different filter stages. When we are increasing the titer, we're changing a little bit the traditional batch process, right? Usually the batch process time in capture is defined by the loading, because usually when we have low titer, you spend a lot of time loading that column to an efficient max loading binding capacity. When we increase that load, then our loading time is much smaller, our cycle gets much shorter, which means we can effectively run more cycles in the same amount of time. However, there's always a caveat here. If we have, we still need that to be cost effective, right? It is not the same to have a smaller column and run 15 cycles than to have a larger column and run less cycles. We need to find a compromise between column size and number of cycles that we required. And at some point, when we talk about very high titers, going above 10, like towards the 15 and the 20s, we then start talking about very large columns, which means that our elution pool, the elution peak that we gets into the 200 to 600 liters. This is volume that I need to move downstream. And I will not have a single cycle, I will have multiple cycles. I can reduce my column, but then I will increase the number of cycles. And then I'm starting to make an argument to move from a batch capture to a continuous capture. Because if I'm going to have more than 5 cycles, and this is my rule of thumb, some people may have a different one, in a batch process, I might as well go to continuous, because I'm already going to get that complexity of having to process many cycles down the road, right? I don't think it would be wise to wait and pull everything. The volume will be very large. I will have very large capital, meaning very large tanks installed, which they vaulted to the floor, and they're not movable. And for commitment issues, I prefer things on wheels that I can move around easily. And it makes the floor space a bit more flexible. So if I'm already going to have that complexity of having to deal with many cycles, I might as well put a continuous capture step, which will require smaller columns, will make my room more flexible, and will give me a step that is more cost effective. And even if we're just talking about a 20% increase in the binding capacity, I'm reducing my volumes by 20%, both in the buffer, but also in the product, which may mean that downstream, that 20% reduction or that minimal reduction makes my equipment a size smaller. I don't have to size up to deal with that. So you can do continuous capture after a fed batch.
Mm-hmm. Sure.
Nicholas, do you have anything to add to that?
No, definitely. I think it makes more sense, like Ruth says, when you get into higher volumes, higher titers, definitely have a look at the multi-column chromatography like PCC in order to see if that can help you with the bottleneck that you may experience trying to process these large volumes. With the better utilization of the binding capacity of the resin and the possibility to run more efficiently and I would say primarily also run your non-loading steps more efficiently. They become an important part of your cycle. That will help drive down your costs.
Yeah.
Any other comments from you, Andreas, on that? The downstream implications, perhaps. Do you have anything you'd like to?
No, it's so obvious that upstream and downstream are connected, and that the titer that comes out of the bioreactor is really dictating the strategy for you guys.
Great. So perhaps we can move on to the next 2 examples, which we've dipped into continuous in the downstream, but perhaps we can look at the next 2 examples in the upstream.
Sure. And we have so far spoken about high titers. When looking into the continuous process options, we actually will deliver lower titers. But let's start with the first option, which is a dynamic perfusion, where we as upstream people actually made our life a little easier. Which means we are not controlling the cell densities. There is no steady state, no steady viable cell density. But the principle is that the perfusion rate is ramped up together with the increasing cell number until we hit a cap, for example, 1 reactor volume per day, and then we maintain that until the culture viability starts to drop with the kind of normal harvest criteria, which is typically after, like, let's say 2 weeks, 14, 15 days, something like that. The consequence for the downstream is that neither the flow rate of harvest nor the titer will be constant. So it will be a little bit challenging and pushing the complexity a little bit downstream. Whereas the other option I like to talk about, which is the steady state perfusion, where we are putting a lot of effort into maintaining a steady state, maintaining a controlled viable cell density, let's say at 100 million cells per milliliter, and that allowing us to run the process basically for 14, 21, 28 days in steady state, and delivering a typical harvest titer, which corresponds to maybe 1/5 of the fed batch titer, but delivered continuously over a longer period. Also with the flexibility of defining batches, and maybe we come back to that when we talk about regulatory aspects, that you could collect harvest for 7 days, 14 days, 21, 28, and have each of these kind of 7-day periods defined as one lot, with the flexibility of adapting your production capacity basically to the market needs, or switch to a new product. So these are the dynamic perfusion and the steady state perfusion are the two that I envisioned to discuss today.
One thing that I like about perfusion is no clarification.
Sure. Yeah.
So that makes it easier for us. Not just because I'm getting a continuous flow, that means that I'm going to do a continuous capture, right? So if I have very low volumes, so my bioreactor is small, I have low reactor volumes of VVDs per day, I could do a batch capture, right? It's small. This makes no sense to complicate anybody's life if what I have to treat is a small volume. If the volumes increase, then the case for a continuous capture increases, because otherwise my intermediate storage will start increasing, and that is called storage. It's expensive storage, both in energy consumption, but also on footprint, which is also energy consumption in the end.
Yeah.
So if I'm increasing the volumes, if I have dynamic perfusion where the flow rates change and the titer change, am I not, in the same thing for steady state, right? I might not treat all the material that is coming, because it might cost me more to capture, purify it, than the money that I will be throwing away by not collecting those first days harvest that has very low titer, right? When we have changing in flow rate and changing in titer, we still need to make sure that whatever comes out of the capture step has a stable profile. So the rest of my train, at least, is one size and is operated the same thing regardless of the day of the harvest. When we have a perfusion rate, they're usually not very large flow rates, right? Which means that if I'm doing an alternative column capture, which still continuous capture but not multi-column, I can, because I'm having a long residence time in the end with a low perfusion rate, I can exploit more of that capacity. So then the case for continuous capture is slightly less attractive, because I can have a higher dynamic binding capacity when I do alternated columns. I will need to change the times, right? Both if I also write continuous, if I get lower flow rates or lower titers, I will need to have my loadings on them for longer, just to make sure that the columns that I have get equally loaded regardless of the day of harvest. When I have a steady state, that is simpler, right? I don't have to adjust for flow rates or anything like that. And I have a stable, so there's no change in strategy depending on the day. Same thing, I can use alternating columns, or I can use a multi-column. Alternating is simpler, but it might be that in order for my process to be cost effective, I need to move to a continuous capture, just because my breakpoint is a little bit more refined, and I do really need that increase of extra capacity that a continuous chromatography system will give me versus alternating columns. Then, now that we touched upon the capture for all of the upstream, we move into downstream, which after capture doesn't really matter, more or less what you did. So if we have batch, and we're not going to talk a lot about the traditional, I think, in order to intensify that traditional batch with low titers, a good idea will be to use single pass TFF up front of the capture. So then I can reduce this volume, concentrate, and I can make my capture step faster. So just getting that increase in titer, not in the bioreactor, but outside of the bioreactor and before the capture. When we are in high titers, which means many cycles, or in perfusion rate, which also means many cycles, I need to start keeping a cadence downstream, right? So I decide which cadence I'm going to keep. I'm going to treat every elution peak as a single batch. So I'm going to keep the cadence of, if I run 150 cycles in my continuous chromatography system, and I'm going to keep 150 cycles going downstream, or I'm going to pull at some point. And this is a purely economical decision. There are steps that will be most cost effective at a certain scale, and will not be so cost effective if I reduce the scale or if I increase it a lot. So there's a break point in some. So it is, for example, we might want to batch after the polishing step. And definitely if I'm keeping a cadence, easier will be to have all my polishing step in a flow through mode, right? There's some challenges of what to do after virus inactivation, because I need to keep them in the right pH and in the right low conductivity, which sometimes is a bit of a challenge when I'm adjusting my pH by adding a concentrated base. So there's some things to try there. One thing that will be really nice is to reduce the two polishing steps to a single flow through polishing step. For me, that's the most beautiful way of intensifying. When you remove the steps and make them, not only remove them, but make them simpler. And we did have some work with the Capto Adir to do that, and it seems that it gave very promising and very nice results in both aggregate removal, whole cell protein removal, which is what you would do with the cation exchange, and virus removal, which is what you would do with the anion exchange, the second polishing step, or alternatively it could be the first one. And then virus filtration, concentration, that can work very in the same way as a batch process, depending on where you pull. So not much complication down there.
Thoughts on that, Andreas? Lot to take in there.
Yeah. Now you're putting the upstream person on the spot, but what I hear is really that continuous can make sense in the capture, but then when it goes further downstream, it's more like a batch-wise.
Because it's not truly continuous, right? The moment that you capture, you move to a discrete process, because your elution peaks are discrete. You don't get a continuous elution flow. So then it allows you to play around with the cadence, whatever is easier for you. I mean, there are customers that do fully end-to-end continuous, down to inline concentration and inline diafiltration. And that is also okay. I mean, you start your bioreactor, you process your entire bioreactor, and that's your batch. It's not 7 days or 14 days. You can decide my entire I have everything so under control that the bigger my batch, the less work I have to do to release it and to process it. So you can go all the way. You can also start batching in your polishing step, or you can start batching before virus filtration, or even after virus filtration, right? We now have filters like the Pegasus Prime that can cope with starts and stops, which usually virus filtration doesn't like. And in a continuous, you need to have the ability to stop and start, because you might not have a continuous flow, because actually it's a cyclic process.
Yeah, but perhaps to consider with a steady-state perfusion process is that you have a very large volume of fluid at low titer coming into the capture step, but then you have a concentration. So what comes out of that capture step really is a very highly concentrated product in very small flow rates coming out of that. So if you start with, say, 500 liters a day coming in, you may only have like 20 liters a day through your polishing steps. So that is why it often makes sense to pool after the capture step of virus inactivation.
It's this economical thing that sometimes a very small batch is, you don't have the economy of a scale there.
Going back to the upstream, we talked about the first two examples. There were some considerations for barriers to implementation, or how easy it is to implement. How about for the two we just discussed there, the dynamic perfusion, steady state perfusion? You said dynamic is relatively easy for the upstream guys, but causes challenges downstream. So what about the steady state? Seems to be an interesting sweet spot, right? Potentially for the downstream.
Yeah, absolutely. I think at least the perceived barriers for process intensification are the complexity that is added, and that is why I pressed when it comes to batch processes on the high seed variant, because it gives you a good gain without increasing the complexity, and that is why I lifted the dynamic perfusion. At least from an upstream perspective, and I think it's manageable downstream, because I see many companies implementing that concept is also very attractive, because you don't need to control cell densities and basically take down the complexity a little bit.
Understood. Any final comments on those 4 examples before we move on? Are we all happy?
Yeah, I mean, I am not the one to run the bioreactor, so I am happy.
So we began by looking at some of the drivers, like the why, I suppose. We've looked at some examples. Now I thought we would move to other areas that may be right for process intensification. So perhaps, Nikos, you can kick us off with the fluid management aspect that I know we wanted to cover off.
Yeah. That's a really important aspect that is sometimes a bit overlooked. It's more interesting perhaps to talk upstream and downstream, but fluid management is super important, especially with intensified processes where you may have tighter scheduling. You need to have these fluids delivered on time, in volume, all the time. So what we see customers doing is they're trying to cope with the large volumes of media that is to be prepared for perfusion type operations, and also large volumes of buffers, especially when you get into high concentration fed batch processes. So the options you have is really, the traditional buffer preparation process, for example, is you do that in-house, you add salt or stock solution, and mix that. If you have, for example, a product that is moving to a new clinical phase, you may need to scale up your process. We have a lot of customers coming to us for pre-made buffers to help alleviate that bottleneck that is generated. It's really large volumes of liquids that are needed, that may be surprising to some customers. So you can turn to an external vendor to source your buffers until you have built that capacity, or you can use that as a permanent solution too. But also, I think it's interesting to look at automation in buffer preparation, and you typically have the options of inline dilution, which is using stock concentrations of concentrated pre-made buffer that you dilute with water to have a more efficient storage and handling of those stock solutions, and be able to dilute that on demand. Or you can look at inline conditioning, which is using individual component solutions in order to mix the proper buffer from a limited set of stock solutions, which is even more efficient many times. So that can help you drastically reduce the amount of floor space that is used just for preparing, storing buffers. Your warehouse area to store the raw materials for this can be reduced many times as well using concentrates.
Well, you said this is something that's less considered. Is it simply because it's not deemed to be as important, or it's just a
I think it is just as important, but I'm not sure why that is actually. It hasn't been the same focus on solutions management.
Never the same focus on the main processes as in the secondary flows, right? There's a lot of focus on getting the process flow right, and there's a lot of work put into it. But then the materials flow, which will be all the buffers and the media, but also all the consumables that you need to manage, that also needs to get consideration because everything that you use for production needs to be stored in the same conditions that is used. So if you don't manage that flow, then you might end up with very high storage areas, which are costly and have to have intense preparations team, either for equipment or for autoclaving consumables, or just for restocking your Kanban or whatever is that you use for storage of your materials, but also for buffer or media preparation. I mean, ideally, you don't want your buffer preparation to run 24/7. Should be more of a relaxed 8/7 or 16/7, right? But when, as Niklas said, when we move into a constant flow, there's also a constant flow of materials and a constant flow of water with stuff.
Water with stuff.
Exactly.
Maybe that's what we need to call it to get a bit of energy. Water with stuff.
I think it's very much like if two process scientists meet and they challenge each other. How many grams of product per liter bioreactor volume a day are you producing? And then they are challenging each other.
Right.
When two plant managers meet, they have completely different metrics. They ask about what is the floor space per gram or per kilo or whatever, and the fluid management speaks a little bit to that aspect.
Also, we tend to talk about specific productivity for the perfusion bioreactor. We forget that the volume that we use is not the volume of the bioreactor, right? Because if we exchange 1 to 2 bioreactor volumes per day, and we run for 15 to 30 days. I mean, exactly. The volume is massive.
Yeah, exactly. We spoke about buffer prep, but media prep is as important. And looping back to my initial comment on the kind of performance of the media platform, I spoke about cell concentration based feeding. It's called CSBR, and there many processes are operated at a CSBR of 40 picoliter per cell and day, but we have demonstrated, for example, being able to get the same productivity at 10 picoliter per cell and day. That's a factor of 4. If you're operating a 1,000 liter bioreactor at 1 reactor volume per day, it's a difference between 1,000 and 4,000 liters of medium that needs to be made every day, and prepping 4,000 liters of medium is not very
this is
fun.
why
and then pushed through downstream as well, through the capture step.
It is important to have the end in mind, because sometimes when we look at things that are very small, right, we want to get it super good, max productivity, max yield, max purity, And at very small scale, 1 milliliter more or less doesn't really matter, but that milliliter or that 100 milliliter will transfer in a 10,000, 1,000 volts downstream to the commercial. But also, like, the materials flow is also something worth looking at for traditional batch processes, right? It's not something intensifying how you deliver your buffers and how you deliver your media is not exclusive to an intensified process. It is something that you should look at in order to make your plant a bit more efficient, because it's like, as we say, moving water up and down and steering water. If you can steer, instead of 600 liter, 60 liters, if you use stock solutions, right? And you reduce the amount of stocks you need to make versus the 1-1 buffer, then your footprint in your buffer preparation area is going to be reduced also significantly, as well as the staffing, how many manpower you need to produce those.
And you mentioned earlier the sustainability angle. Surely this sort of feeds into this conversation as well. This is not just volumes, right? This is energy. This is consumption of material.
Yeah.
So perhaps we can touch a little bit about this newer driver, as you described it, Nicholas, of sustainability. Where are we at there across the whole piece, would you say?
I think we are at different stages depending on where you look in the industry, and some, especially the big biopharma, have embraced sustainability, I would say, at the moment, and also a lot of CDMOs. I think that is a differentiator to the CDMOs, for example, being able to say they have a sustainable operation in the process. So a lot of focus has been on carbon footprint. That is something that is on top of everyone's mind, I would say. So that, I would say, is the key focus area. But also, like, water consumption in some areas is important, and also other considerations. But if we go back to carbon footprint, what typically drives this, if you have a sort of standard energy mix in your facility with quite a bit of fossil fuel producing that electricity, for example, or energy that you need in your facility, then the floor space is super important to reduce in order to reduce the energy footprint and carbon footprint of your process. A lot of the energy is consumed in the HVAC, the ventilation and heating, cooling of your facility, and with smaller volume of your facility, with lower classification, if you can reduce the classification of your facility, not having to push so much air through the facility, that will also significantly reduce the carbon footprint of that facility. But then in some areas, like in Sweden, where we have quite low ratio of fossil fuel in our standard energy mix. And you may have customers in other regions like that, or customers who have transitioned to renewable energy. And when you do that, much of that carbon footprint shifts from being energy induced to being induced by things like the consumables used in the process, for example. So if you have reduced the energy footprint, start looking at the consumables and how you can reduce that. And you can do that in new processes by using more efficient raw materials, perhaps by using more environmentally sustainable raw materials. But also you have the problem of your existing processes. You need to put pressure on your vendors to also start working on the emissions from the production of the raw materials that come into your process. And that is something that we definitely have a lot of discussions about with our vendors at Cytiva and our colleagues too in the business. So that's something that we work a lot on at the moment, because we see this need, especially now, but even more so in the future, I think, to reduce the footprint of the consumables.
Great, thank you. Any comments from Andreas or Ruth on sustainability?
No, only that it's basically on the agenda in every meeting when we meet customers, discussing that. Firstly, and secondly, that it's not that easy. Like you said, where is my energy coming from? That impacts very much the carbon footprint. And I think we are still in the process of putting the map of how do these different intensification solutions that we have discussed actually impact the carbon footprint or sustainability profile. We understand that we can, just speaking about smaller bioreactors, smaller suites, but we also know that we, on the other end, lose a little bit on increased media consumption, water consumption. So I think that there's still a lot of work that needs to be done to get to a holistic picture.
Yeah.
Thank you. Finally, for this section, we also touched upon the sort of regulatory impact of process intensification. I think you mentioned it, Andreas. Can we spend a little bit of time discussing the regulatory implications, if we dare?
Yes. For me, and please chime in, it's a little bit like a perceived barrier, speaking about regulatory implications or obstacles. What I hear from the regulators is that they are very much empowering and supporting process intensification and continuous manufacturing, based on the fact that product quality is better under control, that we expect prices to go down and increase accessibility. So I think we have the regulators on our side, and there are a number of guidelines out there that kind of give a good guidance. For example, on the point of defining a batch, right. I gave one example of like running a steady state perfusion and defining a new batch like every seven days. So it can be time based, it can be raw material based. Every time you start a new media batch, you can. It's really up to it's based on the science and the data that you have produced. People ask a lot about virus inactivation, and I think also there's guidance and
there's not much difference in virus inactivation really, if you can use very similar approach to the batch process. So the Cadence VI system that we have is alternating tanks, but it is same as a batch process. In a sense, anytime that you design a system around a unit operation that is part of your validation strategy, like the virus inactivation step is, you need to consider, is my design ensuring the log reduction that I need to get here? Is ensuring that because I'm moving to intensification, I don't have carryover lot to lot or batch to batch, that I am not the remaining material from lot to lot, if any, is not causing significant impact on the next lot. So there's more work for us to be done in order to ensure that that process works in the new setting, right? Because as I said, it's a different strategy. So there's some things that need to be changed. The same thing for the virus filtration, right? We normally run it at constant pressure and is continuously run, and that is how it's validated. If we move now to different settings, we're not going to run a constant pressure, we're going to run a lower flow rates, we're going to start and stop. We need to add that to our validation strategy, and regulators will ask our validation strategy to mimic real life. I think if you start from zero, I think it's the same work. Very similar work than for a traditional batch process. You might not be as acquainted with the strategies, and this I think Cytiva can be of big help here. If you are, like we said at the very beginning, changing a process that you already filed, and suddenly you require higher demand to meet higher demands, and you change, then yes, there's some things that you will need to refile depending on which steps you take into intensification. how different is your new intensification strategy from the previous one? There's regulatory filing if you change your column diameter, right, or your resin. And now, since COVID, I think regulators are a little bit more relaxed on changing filters, changing resins, and all these changes have, as I understand, have become less demanding in terms of the work that you need to do to file them. And I'm expecting the same thing and clarity coming from all these new regulations that have been in place to support filing intensified and continuous processes.
Yeah. No, I think so. I mean, I see a lot of support from regulators. It's a lot happening in that area, like you mentioned, new policies and guidelines around intensified manufacturing. That said, I mean, their mission is to ensure that the robustness of the processes that we have and the safety of the product that is produced. And we need to have the same demands on an intensified process as a traditional process. And sometimes doing things slightly differently, that means there is new space to explore, how to prove that you have this robust process, this safe process, which can be a bit challenging. But otherwise, I think don't be afraid of those challenges. Have a discussion with your regulatory partner in order to find solutions, because I see a lot of willingness to support in this from regulators and also from vendors like Cytiva. We are here to help in these topics too.
Great. Well, that's a good segue into the final part, I suppose. You talked about the regulatory aspect being more of a perceived barrier than a one in reality. So what are some of the other sort of perceptions that you see from biomanufacturers? I suppose the one that springs to my mind would be that process, and indeed the title of this topic is intensification, not complication, but I suspect one of the main perceptions from biomanufacturers is the introduction of more complexity. But I'm also assuming that Cytiva is busy working on solutions that reduce that barrier. So can we spend a little bit of time on biomanufacturer perceptions and how your conversations typically run with customers? Should we start with you, Andreas?
Yeah, I mean, we spoke about the complexity and weighing different options against each other to end up in a spot where you feel comfortable with your organization. I mean, it's also a matter of training the personnel and having the skills that is required. It's a matter of creating an infrastructure with respect to digitalization. You mentioned that in the intro. All these things are topics that we discussed with customers and look into the specific cases and find solutions together with the.
Because I think you said, Nicholas, that really every customer is in a unique situation, right? They have a different demands, different needs, and so on. Yeah. What are your sort of big take homes from conversations with customers? What sort of questions do they come to you with when they are considering the why of? I guess they've got the why, right? Yeah. That's almost obvious.
Exactly. Yeah.
They have certainly questions.
We're at
about
the what?
Yeah.
Yeah. I mean, a lot of customers come and have a perception that a fully continuous process is the ultimate state and most cost efficient. I would challenge that and say that is not always the case. And especially as you start taking into considerations implications around if you have to transition to a new way of manufacturing, etc., if you have existing infrastructure. But it's definitely an interesting way. But the first complexity is perhaps to understand which technology is the best for my individual needs. Both in terms of cost and also the management. I mean, we've been talking management of raw materials, regulatory implications, etc. And if you have a product that you expect to have a low demand. It may be very potent, it may be used in small doses, it may be for a rare disease. It's typically less interesting to really push the intensification to a super efficient process, as that often means you need to spend a lot in development and may end up with a more complex process. Perhaps it's more efficient to have a slightly simpler process. I think simplicity is something that is sometimes underestimated. And I smile when you say that you think it's beautiful with the simplistic process and taking out process steps, because I think that's a super powerful way of intensifying the process. That said, when we have large volume demands, there are many cool technologies and efficient technologies and new generations of raw materials that can be used to make this more cost efficient, more productive, increase the throughput, etc., to help getting to a lower price point when it comes to manufacturing costs, to increase accessibility of these sometimes rather expensive drugs.
Any final comments on that?
Yeah.
What do your customers talk about?
So it's always perceived as more complex technology, more complex to schedule, because you have, as we spoke, many more cycles to schedule rather than today schedule one step. Tomorrow think about scheduling the next one, right? You need to get a good scheduling strategy there. I do not think that the increased complexity, which in my opinion is also coming from the data flow that we have barely touched upon it, right? All the data that is coming out of there's a continuous flow of data. But I do think that's getting more complex also on a batch process, because we're getting access to better tools. Better tools get us more data. More data needs to be made meaningful, needs to lead to meaningful decisions, to something that is not just numbers, right? I think we're coming to a shift in every kind of work where the skills that are required are slightly different than they were before. So, and we all in our work need to become more and more often comfortable with the uncomfortable, with new things. And that is what happens with intensification. That's what I say, is doing something different. You're going to do it differently, so you need to switch. You're no longer doing your risk assessment in the same way that you do for the batch. If you apply that line of thought, then your risk profile of an intensified or continuous process is going to be very high. And that's, I think, sometimes is perceived risk, because those systems are designed to manage that risk, right? So it is being a little bit more comfortable with the unknown with new things. And definitely the digital part of the industry is growing very fast, and that also requires different skills.
I think you made a very good point saying that there is not only fed batch and continuous process, that there is something in between, that or the intensification variants that we spoke about, connecting the process, and then in the end there is a continuous process, and it's really up to each case to understand what are the bottlenecks, what are the demands, where are we in this scale, and what can we work on to find the best solution.
And it can be done, right? Do we have examples?
Exactly.
Obviously, we can't talk about specifics, but perhaps we can just spend a moment looking at the sort of success stories that you've seen in your time with various customers on where they've achieved, the goal that they set out to achieve, and perhaps a little bit about, the approach they took. Is that something we can share?
Yeah, I mean, just looking in the literature, there are a number of cases speaking about high seeds fed batch processes, and describing what I mentioned earlier, the gains in overall productivity, in higher titers, and that is something that I see, like I said, regularly implemented from the start for new processes, even second generation processes consider that option. So that is really a low hanging fruit. Also in the public domain are kind of companies like AstraZeneca developing their next generation a platform which will be built on perfusion, a dynamic perfusion. So I think without revealing customer details, there's a lot of information in the public domain highlighting or supporting very much what we have discussed.
Yeah, and I think we have probably some webinars from the past still on our website. For example, if you want to see some examples of customers who have been sharing things like improving the buffer management, moving to continuous processing, etc. Search for process intensification on the cytiva.com website.
We'll make sure those resources are available to you.
Perfect.
Finally, then, I thought, we've talked a lot about the now and where we're at, but I think, as with many things, there is not a single point at which we've achieved, the totally intensified process. So I thought it'd be good to cast our mind's eye to the future, just to think about, how process intensification evolves from here. Maybe Nicholas, you can kick us off.
Yeah.
Where are we going next? Again, without revealing too many secret details.
I think to some extent we're moving into this gray zone with combinations of batch and continuous processing to optimize the process for different purposes. That I see a lot of interest in, I would say. And then also cell line development, really important in order, like we discussed, to bring up the titers. That will significantly help the productivity and economics of your process. We see, I would say, an increasing interest in perfusion in the business today. So that is growing, even though fed batch is still the dominant in the industry. And also, I would highlight perhaps the capacity build out of also the very large stainless steel facilities that by some may be considered as sort of things from the past, but we see a lot of. Doesn't have to be too many of these to have a very large part of the global capacity. And the reason for building that is because of the economy of scale, that you can produce things very efficiently, very cost effective in very large steel facilities with several bioreactors feeding perhaps one or two downstream lines.
Okay. I am more of a hardware girl, so to me, I think we need to move more. And I see also that in discussions, flexible platforms. So equipment and platforms that can today do one molecule and tomorrow another molecule tomorrow. Today they work in a setup and tomorrow we shift around and the setup is different, right? And that is the opposite of the large stainless steel installation. So there's room for like a high variety of strategies within the industry. As I say, commitment issues. I like small things that can move around. I like to be able to exploit that flexibility that, for example, single use brings to the table versus stainless steel. I also think we're moving towards heavier PAT, inline, online, align, all that detection methods to just harvest all that data and make it meaningful and try to feed it back into the process so we can keep a process deviation free. And also we can apply on-time release, on-time batch release and have like very fast decision making.
Andreas?
Yeah, I'm thinking about the point I made a couple of minutes ago about the evolution from fed batch to continuous. So on the process side, that is my picture. On the analytics, more PAT, but also a kind of intensification of QC methods in a high throughput matter. in moving towards multi-attribute methods, going to at-line methods, and then in the end having an inline method which basically keeps the process in control and avoids deviations. And then finally, another bucket is the digitalization, which we have also touched a little bit on, where we are now in a situation of having kind of silos between unit operations, not being connected and communicating with each other. I think that is also something which we are seeing heavily, that we get a unified structure so that we can make use of all the data from PD into manufacturing, build digital twins, and have a kind of an adaptive process control based on all the available data.
Fantastic. Well, I think that draws our discussion. I wouldn't say to a close. I feel sure when the camera shuts down, there'll be more discussions to be had. But for now, thank you very much to our three experts. Phenomenal, Andreas, Ruth, Nicholas. Really appreciate your insight. It's fair to say I couldn't have had this discussion without you. That would have been very strange indeed. And thank you all for joining us and watching. Goodbye.
Bye-bye.
Bye.
Bye.